Okay, we're a little over schedule, so we're going to skip questions for now, if that's okay, and move on to the next talk. Our next speaker is Dr. Batta-Lona, who is an assistant research professor of marine sciences at the University of Connecticut, where her research focuses on mesopelagic faunal diversity and trophodynamics. So please take it away.
All right, thank you for being here. So this is great. I'm going to continue with the web analysis and integrative techniques to do so. So today I'm going to talk about salp and fish diets in the Ocean Twilight Zone. And this is work that I'm representing for a bunch of groups, especially the Joel Joppies group and Simon Thorold. So we're going to try to provide new insights from DNA barcoding, microscopic analysis, and stable isotope analysis. So first, the integrative analysis of the mesopelagic food web. I hope to provide a novel look at mesopelagic trophic pathways with a focus on fish and salps. For that, I'm going to use I'm going to present data from molecular metabarcoding and morphological microscopic analysis, especially for fish. And hopefully, as we've seen with other talks, highlight the importance of gelatinous organisms in the prey components of fish gut content. I also would like to touch a little bit on hopefully try to understand the sources of this productivity using data from the Simon's lab, using isotopes to try to understand whether if it's coming from the surface or the deep detritus. So as I mentioned, my talk will be divided into two organisms, the mesopelagic fish diets, where I'll be comparing metabarcoding results and morphological results. And for the salp diets, I'll be showing some metabarcoding results for 3 salp species and some isotopes results as well. So both fish and salps were collected in the Northwest Atlantic slope water. Here I'm showing the maps of the collections during 2018 and 2019. The cruises were on the Bigelow, and they were in August as well in 2019. And here I'm showing the gear. Obviously, we know about the biases. So there's some samples that came from the Magnus and then some samples that came from the midwater troll. So metabarcoding, we've been talking about the eDNA and metabarcoding. So I just want to orient you on the markers that we're using. We use metabarcoding with 18S ribosomal RNA. And this is called the tree of life gene. And as you can see, it's found in all living organisms, all the way from dinoflagellates, salps, copepods, all the way to gastropods. And the other plot that I'm showing here is the full length of 18S has the different regions. And just like we've been hearing about the nets and how they're biased in what they catch, these different 18S regions can also be biased in terms of what organisms they can identify, sequence, or we can classify. So for the hypervariable B region is the most widely used region, and this detects organisms of many taxonomic groups, but the level of identification will be a taxonomic group. Like it won't be as specific as B4. This other region B4, it's able to classify to a species level for certain groups. So like I said, B9 will give us a big overview of multiple plankton groups, and B4 can allow us to focus on certain groups to the level of species. And now the other marker that we use is CO1, and I think we've been hearing about this marker both from Census of Marine Zooplankton and other efforts. So the advantage of this is not only that it's found in a lot of the organisms that we're going to be sampling, but it also tends to discriminate both in the group level and the species level. And like I said, because we're able to go to the species level, this can allow us to do analysis of biodiversity at this species level. For both of these markers, we use the reference barcode database that are available, and I will talk a little bit more about these databases in my results slides. So here we have all of this. Like I said, CO1 can have high affinity to sequence or to amplify certain groups like copepods, and this is the overview of all the different groups that CO1 is able to identify. Now using these markers, we apply it to metabarcoding of diets, right? You can use it for eDNA or for other applications. But in our work, what we do is that we start with the DNA extractions for digested gut contents. So the gut contents will have a plethora of all these taxonomic groups. And then once we have the DNA, we do PCR, which basically we're making tons and tons of copies of the markers that we're interested in, which are short gene regions of the hypervariable region B9 and B4 of 18S that I just showed you, as well as CO1. Once we have all these multiple copies, we send it to a center where we do high throughput sequencing. So basically we sequence everything that we found in, whether it was a fish gut or a Salp gut. And once we obtain millions and millions of sequences, we use bioinformatic pipelines. In our case, we use Mother for all of our markers. And we use, during these pipelines, we use the reference database that allows us to identify and classify these sequences. And now jumping into our results, these are the results for the V9 metabarcoding analysis that we did and the microscope analysis of fish diet. Here are all the 5 fish species that we looked at and compared these results. These plots that I'll be presented throughout my presentation have the relative abundance on the y-axis, and on the x-axis, I'll have different things that I'll be comparing. But in this case, I'm comparing the metabarcoding data identified with this icon of the whole double helix, and then the microscope for the microscope counts. So we can see all the groups that are detected that metabarcoding are a lot because of that ability to even able to detect things that are not distinguishable with the eye. And this is all the microscope counts were done by the Jopis lab. And we see that they correspond, some groups really correspond, right? Like amphipods, other crustacean, and copepods. But then when we come to the yellow section of the microscope analysis results, then we have the soft body, which we know already, you know, some things you're able to identify like cephalopods because of the beaks and stuff like that. But other things are gonna be highly digested and it will be hard to identify morphologically. But metabarcoding will kind of fill the gaps. So for example, for the hatchet fish, this yellow section, now we know that actually some of it might be salt, some of it might be bryozoan, siphonophores and ctenophores, and so on and so on. So B9 sequence proportions indicate relative importance of these prey groups, and the microscope counts kind of correlate and also gave us a relative abundance of the prey. And that was B9. So remember, B9, it's all about big groups, right? General groups. But now when we go back to CO1, which identifies species accurately, especially for crustacea, here I'm showing the same fish species. I think there's one more than I didn't have in the comparison, but I won't even attempt to say the scientific name because of my accent. But also we have, for example, that some of this data shows that for Igofumigomi, for example, we can see that the different prey species by group are over here, has a lot of the composition, a lot of the preys are part comprehended, comprehended by copepods up here, right? And then when we move farther down, we see that NS, the NS species, which is this fish, prefers this kind of ophocytes compared to the other fish species. The rest of the other fishes prefer Meganictiphonus novergica and Nematoceres megalops. But we also see that there's Digmons elongatus has cephalopods in their diet. So we're able to identify different things now to the species level with CO1. And I was mentioning about these databases that are really important to be able to identify all these prey components. And these are the one that we are using is Metabarcoding Barcode Atlas for CO1. And I think Heather yesterday was going to get into Metabarcoding, and I highly recommend this database because it's a compilation of gene bank, and so it's a curated database for markers like CO1, 18S, 16S, and I think now soon 12S. So with CO1, we see that the predominant prey species differ among the fish specimens and also the fish species. So now with this information, we can start tracing it down into the food chain, right? Because we are able to identify those pre-species. So now with behavior, distribution, and the diet of predominant prey species can allow to determine the source of productivity, whether it's the surface or whether it's the deep, right? So we have a different species, and then it would take into account, are they diel vertical migrated? What are the dish that the diet that we're finding, and what is the distribution? And the other thing we can do with metabarcoding, similar to what Anola presented, is that we can trace it, use network diagrams. So here I'm showing the predators on this side of the plot and all the prey, and we can see the linkages between the 7 fish and the 14 prey groups using BeatMap and other programs. And we can see again that ophausids is a strong component of their diet, but we can also see that the jellyfish are have a definitely wider link, which means that are important for certain of the fish species. If you want to learn more about this, please visit Anne Buckland's poster. I think she's in the JETZON session on Monday, I think. Is that where it is? Okay, well, you can stop by and look for her poster. So now going to the salps and the mesopelagic food webs, we've seen this picture before. Here at the salp, belonging into that, that ecosystem. And salps gather, let me see. Can I make the video work somehow? Oh, anyway, I guess I'm doing my representation of a salp swimming. So salps gather food by pumping water. So this is a picture. This was supposed to be a video. This is salp at Thompsonite, and they have an oral aperture and an atrial aperture. So basically, it's just a tube. For some of you, if you're not familiar with salps, I know Alexis is, but they tend to like swim in the water column by compressing their body in their muscle bands. There he goes. It goes like this. And so while they're pumping water through their body, they have like a mucus mesh inside, and all the particles get trapped in that mesh, and that mesh eventually makes its way with ciliates all the way to the stomach, which is here in the orange. So because of this swimming behavior, they have high filtration rates, and they can adjust very quickly to fluctuating food concentrations by filtering a wide range of prey sizes. And because of, I think Alexis mentioned, they have a really complex life cycle, so they can alternate between sexual and asexual reproduction. So that's why sometimes they're very annoying. They have blooms and they clog your nets because they tend to do this. So some of the questions that we want to find out is, what are salps feeding on? Is it always the same diet? Stay tuned. Just a quick recap. They were collected in 2018 and 2019 in the Northeast United States. And when the salps, or when all the zooplankton was collected, the scientists, I think Kayla was on board, they were able to select the salps and separate them and freeze them. So back in the lab, I identified and separated them into the 3 species that I'm going to present today: Yassi sonaria, Salp aspera, and Salpa fusiformis. And then Kayla, I was able to dissect the tissue, the muscle tissue from all of these cell species, and I sent this samples to Kayla, and she carried out the compound-specific stable isotopes here at Woods Hole. And back in our lab, what I did is that I dissected the salps. Like I don't know if you have encountered them, but the outside layer of the salps is very thick. So I removed the muscles, and then I removed all of these orange blobs that we see here. There are their stomachs. So now going back to the sequence results that we got for V9, and this table, I'm showing the prey groups that we detected. I'm showing the different cell species on the top. And for V9, the predominance was not surprising. The sequence is more predominant with salp, so we definitely have some predator swamping. But we also have detection of other groups, right? And among them, the amphipods and the ciliates, which we only detected for V9. But we also observed that dinoflagellates and syndiales, which are parasites, were highly abundant in the prey components for 2019. I'm only showing 2019, but there were similar patterns for 2018 as well. And the copepod sequences were definitely higher also for 2019 with the V9 sequences results. And now for B4, remember, this is also 18S, and I'm just going to show results for all the groups. And as I mentioned before, there's biases with these markers. So B4 definitely didn't detect any amphipods or any ciliates, and these are exactly the same samples and the same species. But there were some similarity and agreements between the markers as well. So copepods were also more abundant in 2019, And of course, we detected a lot of salps from the predator swamping, even though I really tried to wash all the stomach tissue from it and trust to like the stomach contents, we still have some predator swamping. But we also saw some sequences for fish, gastropods, and siphonophores, which we think are probably likely contamination from sampling. So those were removed for downstream analysis. So we only focus on 5 groups, which are the copepods, dinoflagellates, syndinellates, and rhizaria, and diatoms for our downstream analysis. And I want to show you how those proportions, like relative abundance, looked for the salt gut contents. So again, we have relative abundance on the y-axis, and these are the different salt species. So you see that the different prey groups, diatoms, syndinellates, and dinoflagellates, are not particularly different between the species. We don't see any striking differences between the 3 salt species. So our following analysis are only based on these prey groups. And when we did some Shannon diversity analysis for the salt gut context, we see that for both, and these were based on sequence numbers that were log transformed for the prey groups that I just mentioned. Here we have the Shannon index on the y-axis and the different salt species with different colors. Yes, Isonarius red, Salp aspera green, and Salpa fusiformis in blue. And we see that the results show a wide range of diversity values among the salt species. And there were marked differences between the markers for some salt species. So we see that Salpa aspera with B4 has like a wide lower range of Shannon index values, versus for V9, it has a more compressed range of values for the diversity of the prey. And that could be due to the classification that we're able to get to. And you can come talk to me about why those differences might be based on the marker that we're using. So as I mentioned before, the marker B4, the hypervaluable region, is able to detect species. So we're able to dive in a little bit more into some of the groups that we were detecting the gut contents. And here I'm showing, again, relative abundance on the y-axis, the different salt species for 2018 and the different salt species for 2019. And here are the list of diatoms that we were able to detect in the diet of salts. So the most abundant diatoms that we detected was Thales acera in the lighter blue, and then mini discus in the navy blue. And Salpa fusiformis gut contents in 2019 over here have fewer diatoms genus or species. I thought that it was because my sample size was smaller, but it's actually the same sample size as Salpa aspera. So I'm not really sure what happened and how much this is reflected on what we can find in the water column. And Yassis sonaria that was collected in 2019 had the most diverse composition of the diatom species. So now moving into the isotope analysis that Kayla carried out, she compared also the 3 species only for 2019, and she didn't find, or the results show that there were the 3 species show very little separation between food sources. So over here we have the food sources in the different color ovals. So bacteria is over here, diatoms and dinoflagellates, and here you can see the species. Yassis sonaria are the circles, Salpa fusiformis the square, and Salpa aspera the triangles. So there was little distinguishing or separation between the salt species. Not surprisingly, we saw the same in the metabarcoding, right? They were very similar in the diet composition. But we also see that all of the Salpa species group really close to the diatom food source, and there were some that were in between the bacteria and the diatoms. We didn't analyze any metabarcoding data for bacteria, so unfortunately there's a gap there, and maybe for future studies we can start thinking about looking at bacteria, since I know cyanobacteria has been observed in the gut content visually. I think Debbie mentioned that yesterday. And for more details about this analysis, please stop by and see Kayla's poster later today about the isotope analysis for salts. So I hope that with this presentation, I am able to convey that obviously the same as what Anola presented, is very important to be integrating different approaches of analysis of the mesopelagic food web. So hopefully I showed that metabarcoding allows identification of pre-species and tracing back down chain, chasing back food chain to sources of productivity, that morphological Microscope examination allows comparison with early studies of the relative abundance of these prey groups, and that compound-specific isotope analysis complement metabarcoding data by providing a long-term view of the food web interactions. I forgot to mention that with metabarcoding, we have a snapshot, right, of what they ate in that moment when they were caught. But the compound stable, the stable isotope analysis will provide us like what they ate a couple of weeks or months before. So now we can have a better understanding or what the role is. So now the next question is the easy questions, right, Tracy? So what does this mean in terms of the carbon export? So stay tuned. I have another, first off, I want to acknowledge and thank everybody that for the funding that we have from Woodsall, also all the crew that collected all our samples and the sequencing centers and the beautiful pictures that I borrowed from Benjamin and from Paul Kager. And going back to the carbon question, we have another set of samples that we collected in 2021, and this is just Salp aspera, and we have actually depth profiles of the metabarcoding cut contents, so please stop by my poster later today and I can talk to you about that. Thank you for your attention, and I can take questions if there's time.
Yeah, we are back on schedule, so we have time for maybe 2 questions before we have our 30-minute break.
Yeah, I guess we can just leave all the questions. Oh, sorry.
Oh, hi. I have a sort of general question to the 3 speakers. So I was wondering whether, people can use, the predator harvest data, say the tuna, swordfish harvest data to analyze or predict using, regression analysis, the location and the quantity of the mesobiologic biomass. Thank you.
I think that's more of a question for the other.
Yeah, that's a really interesting idea. I think to first order, we probably don't have a good enough data set of predator gut contents that are especially resolved in space and time to do that. That would be my sort of first order response. I don't know. What are you thinking of?
I think that I would agree with that. We're working on synthesizing the lancetfish data set, but it's obviously one species from one fishery, but it's over 15 years, and we're up to about 3,000 stomachs and hundreds of thousands of prey items, but I still think it's not quite at the resolution needed to do something like that, unfortunately.
One more, and then we'll do our break.
Thanks for the talk. I'm interested in the effect of sample size when looking at diets for the salps, the siphonophores appearing in large numbers, wondering whether that was swimming through a patch of larvae maybe or eggs or something and eating that. And in Anila's presentation also, the high level of specificity in prey selection in the mesopelagic, how much that's actually because there aren't that many observations for each species. So it just looks like they're very specialized, but there's only 1 or 2 observations.
Yeah, well, especially for salps, I think there's a recent publication that they could be the eDNA samplers, right? So they're just like filtering all the water. So there could be that there was a siphonovore passing by where they were swimming, and then we capture that. The other thing is that, I think that was another part of your question that I wanted to answer. We had a larger data set with the other analysis that we did with Salp aspera, and we still see some siphonophores. So I think, yeah, I don't know, it's not changing a lot where the salps are in terms of the diet composition, but we do see those rare siphonophores in some of the samples, not all of them. So you're right, it's probably more like a occurrence, and that's why we decided to not take that into consideration for our analysis.
All right, with that, let's wrap it up for now and give our speakers another round of applause. And we now have a 30-minute break, so go forth and drink coffee.
Me back here at 11. Thanks. If you haven't dropped off your poster, make sure you do so.